talked about an introduction to inheritance patterns. We specifically got into the idea of complete dominance, and this complete dominance was founded or grounded in Mendel's laws. And you did your Mendel presentation where we talked about the laws of segregation and independent assortment, specifically saying, that these alleles are segregating or separating. So specifically the idea here for independent assortment, first of all, as a review, the law of independent assortment is saying that if we have, for example, in Mendel's pea plants, if we're taking a look at one trait which might be flower color, okay, and then another trait which might be the height of a plant, the height of the plant. All right, so again, this goes back to the garden with Gregor Mendel. All right, these are the garden peas. All right, so if we had our first trait, which is flower color, we might have purple flowers, which were dominant, so that would be big P, right? And then we might have white flowers, which were recessive, which is going to be a little p. And then for plant height, we had tall, which we denoted with a big T. And then we had short, which was recessive, and we denoted that with a little t. Today we're going to start to take a look at, or in this lecture, start to take a look at dihybrid crosses that are still are still completely dominant, all right, where one of the alleles basically overpowers the other allele, all right. But the law of independent assortment basically is saying that this trait, so if the trait is flower color, this flower color trait is going to be passed on independently of this plant height trait. So they don't have a direct correlation with each other. They are independent of one another. That's very important to understand. All right, but we will be looking today, for example, at what's the chance that we're going to get garden peas that are purple and short at the same time. That would be very important for breeders that are trying to figure out what type of plants they're going to grow, maybe if they owned a nursery or something like that, or we're just simply doing genetic research on plants. So that's our first big law. I wanted to give you a good review and a good overview of Mendel's laws that you guys shared in your Mendel experiment. So that's our law of independent assortment. Now let's take another good look at the law of segregation before we dive into our dihybrid cross. So for our law of segregation, let's go back and we're going to stay with plant height. Let's stay with our flower color example. So we might have, for example, purple, right? So one parent in this example might have the allele for purple, which we are utilizing as big P, all right? But they may also be a carrier for the allele for white, which is a little p. Okay, so I'm doing this to kind of show you the difference between the two, and that's my little p. Hopefully you can see the size difference there. Okay, so we're going to cross that. So we're going to say that this is the P1 generation, or the first parental generation. And we're going to cross this now with another plant, all right, with another flower, and we'll say this one again has the purple, but we're going to say this one's homozygous dominant for purple, so it's going to be two large peas. Okay, so this one again is homozygous dominant for purple. We do this cross, okay, but what the law of segregation is saying is that this allele or this allele is going to segregate or separate from the other, and only one is going to be passed on. They are going to segregate from each other. All right, this is the specific definition then. Only one of these will be passed on, and that's a review there of your law of segregation. All right, so we're just kind of right at the tail end of complete dominance. We did not get to a dihybrid cross in the last example. We're going to stay with the P's, and so basically with the dihybrid cross, what we're looking at is two traits. So we want to see the chances. Well, what are the chances that my dogs that I'm breeding, for example, are going to have a certain fur color and maybe have a certain type of ear, right? Or what are the chances that my horse is going to have a certain type of trot and is going to have a certain type of leg size, for example. So these are all different traits that breeders are able to manipulate. We're going to keep it simple, though, and stay with Mendel's pea plants. So let's take a look at this dihybrid problem. All right. So, again, here's a good example problem. This is the type of problem that you might see in one of my quizzes or tests. We have a pea plant, and make sure you're writing this problem down completely in your sketchbook or written notebooks. A pea plant that is heterozygous for round yellow seeds is self-fertilized. What are the potential phenotypes of the offspring? Okay, so that's what we're looking at here. And we want to always remember our 3 steps to solve these types of problems. We want to follow through with those 3 steps. So our first step is to always identify basically what are the parental genotypes. All right, so we have it's going to be self-fertilized, right? So we're not crossing it with a different plant. That's one of the things with plants is they're able to self-fertilize. So we're going to take the round and yellow seeds. So if it's heterozygous for round, then we're thinking it's going to be big R, little r. And if it's heterozygous for yellow, we're thinking it's going to be big Y, little y. So that's step 1 of to actually write the cross or write what the parents are. We're going to have the first one, big R, and then big Y, little y, crossed with big R, big Y, little y. All right, that's our step 1. Our step 2 for any problem, we're going to actually add a step for the dihybrid cross, which is going to be to foil our cross. So again, here is our cross. And what we're doing is we are taking these traits and basically like putting them in a bag and mixing them up. The ones that I've highlighted in yellow here. We're going to mix those up because we want to see what are the chances not only that we get a round plant, but what are the chances that we get maybe a round and a yellow plant at the same time. Okay, so that's what we're looking at there. So to do that, we're going to go back to our math lessons and we're going to do some foiling. All right, so we're going to take the first here, okay, the first gametes, which would simply be big RY. We're going to take the outer gametes, which would be big R, little y, and we're going to take the inner gametes, which would be little r, big Y, and we're going to take the last ones here, and it would be little r, little y. So this is going to be for parent number 1. So that's our first parent. And in this case, it's a self-pollination. So we don't really have to go through and foil the other group. They're going to be the same. So parent number 2, and we're going to put those here. Now we're going to be building our dihybrid cross. Okay. And we're going to put those on this side, my other ones. There we go. And so obviously you'll want to pause this to check yours, but this is the main idea as this question is asking for the phenotypic ratios. A couple little hints. You're just going to want to be careful as you go through these and make sure that you're putting the right letters in and keeping them nice and neat and ordered. Okay, but you can see there that at the end that we have a 9:3:3:1 ratio, which is a pretty common ratio for a dihybrid cross. All right, so moving on from the dihybrid cross, which really wraps up our complete dominance, it's now time to take a look at the exceptions to Mendel. All right, so these are going to still follow some of the basic principles. Again, It all started with Gregor Mendel, but these are some exceptions that we have to analyze. The first one is going to be what we call co-dominance. So with complete dominance, we had one trait that completely masks or overpowers the other trait. In co-dominance, we see that both alleles, which of course are a variation of a trait, are shown in very close to the same amount. Okay, so if it's in flowers, for example, if there's two colors and it is co-dominance, then both colors are going to show through on the offspring. Another good example of this is with chicken feathering. All right, so with the chicken feathering here, you can see we've got three types of chickens: black feathers, speckled feathers, and white feathers. All right, so in this case, if I was to take, let's say, chicken number 1, I'm going to take this black feathered chicken. All right, I'm going to use that one for chicken number 1. And then for chicken number 2, we're going to use this white feathered chicken. Couple of things. So it's a little bit different in terms of how we actually write the alleles out. Okay, in complete dominance, we just wrote like big A, big B, little b. For this, we're actually going to identify the gene itself or the trait itself. So this C right here is the actual trait, and then the superscript then is going to be the allele or the variation of that. So there's our big B for black feathers. All right, on the white feathers over here, I got C again for the trait, and then it's going to be this big W for white. All right, let's go and take a look at a practice problem here for complete dominance. Take a moment here and get this practice problem written down in your sketchbook or your written notebooks. We're going to cross a black feathered chicken with a white feathered chicken where the feather color is a co-dominant trait. So remember, in the actual question, I will make a note and tell you usually if it's co-dominant, if it's incomplete dominant, or if it's sex-linked. Those are going to be our main exceptions to Mendel. So as you can see here, as we go through and do this cross, we've got our traits and our alleles written appropriately with the superscripts. You go through and you follow the same process, and as you can see with this, we're going to have a pretty standard genotypic and phenotypic ratio here that all of them are going to be speckled chickens. Let's go ahead. We've got our 100% genotypic ratio of CB/CW and our 100% phenotypic ratio showing that they would all be speckled. Our next example of codominance is going to be blood type, and this is an interesting one as we will spend a lot more time on blood type as we dive into evolution, because blood type has definitely evolved in humans. So we'll get into that more, but first I want to talk about how we do a blood type cross. Start with blood type. You will pause this at any time, obviously, get this down. You are going to have to memorize the 4 phenotypes for blood type. You are going to have to memorize the genotypes that correspond with those phenotypes, so you can pause, get those in your notebooks at any time. However, for blood type, there is a specific gene for blood type, and we're going to use the gene is represented by the letter I that we look at with blood type is going to have a letter I in it somewhere. So you can see our first phenotype for type O is going to be little I, representing it as the recessive. This is a great example of how recessive traits aren't always the traits that are found the least in a population. So again, go ahead and get these blood types down, and now we're going to take a look at a cross in a family here for blood type. So here we go. We have our first group up here at the top. This would be the parental group, the P1 group, so mom and dad, and then they have their offspring. So this is our P1, P meaning parental. We have our F1 generation, which would be for the filial. This is the first offspring. So, for example, if this person here was type O blood, that would be little I, and that means that the parents could all possibly have these blood types up here, and you're very simply just using those genotypes and phenotypes that we just looked at. The last exception of Mendel is going to be sex-linked traits that are attached to the X or Y chromosomes. And for these sex-linked traits, here's a great example. So first of all, we've got to make sure we're clear that males have one X chromosome and one Y chromosome, and that females have two X chromosomes. So here's an interesting example. First of all, for our class, we're going to have to be familiar with two sex-linked recessive disorders. That would be color blindness and hemophilia. All right. Color blindness obviously is the inability to appropriately differentiate between red and green color. And then hemophilia, which is a very rare disorder, but it's a disorder that can be managed. There's unfortunately no known cure, but that disorder is going to be found in about 1,000 cases per year in the United States. So again, very rare, but it prevents blood from being able to clot effectively. All right. For hemophilia, we're going to use a lowercase h. For color blindness, we're going to use a lowercase r, because those are just kind of the most common used alleles for this. Again, these are sex-linked recessive disorders, so that means that they're going to be more commonly seen in males, all right? And that is because they only have to show up on one of the X chromosomes in a male. Male only has one X chromosome. So, for example, in this family, we see that dad has color blindness, all right, the inability to differentiate between green and red color. So I've colored in this box here for dad because we know he has color blindness, all right. And then we're going to go down in this family. They had two boys, all right. So both boys received the X chromosome from mom, all right. If they didn't receive the X chromosome from mom and they received the X chromosome from dad, then, well, they wouldn't be boys. They would be XX, okay, which would make them a female. The Y chromosome then was received from dad, which means there's no way that either one of these offspring would be able to receive the little r, or that recessive r, that would cause them to be colorblind. Most likely, in this instance, likely in this instance, mom. All we know about mom is that she's not colorblind. We don't know her genotypes, but most likely her genotype is probably going to be this one right here, simply because it would lessen the chance that it was this one, because that means she could have perhaps passed on that colorblind gene. Since she passed the X on, that would have made the offspring colorblind, and we simply know they're not colorblind. If this was her genotype, however, then this would be what the pedigree would look like. We would color it in halfway like this. And then that wraps up our look at the exceptions to Mendel. It also wraps up our look at dihybrid crosses. So there we go.